Let's see. Is it working right now?
Thanks. Yes, it works.
Oh, cool. That's good. That's good. Okay. Where am I? I have to re-enter the presentation mode. Yep. So we would like to study this nucleus to cell ratio homeostasis problem. So we will use a simplified stochastic gene translation model to calculate the probability distributions of protein numbers. So this is done by solving the chemical master equation corresponding to the model. And once we have this solution of the master equation, we can use this solution, which is the joint distribution, to calculate the nucleus to cell ratio curves. And I will then show some results. But finally, we have this Taylor expansion approximation of this nucleus to cell ratio that helps explain some observations. And last, I will talk about the conclusion and future work. So keep in mind, this is still a work in progress. I can only present what I have. There might be some things that I don't have. I will mention that later. So anyway, so where it started, we want to study the nucleus size control of cells and this NC ratio problem. So we know that cell size can vary between different types, and for the same cell type, different conditions like growth conditions, osmotic conditions, they all affect the size of those cells. But it has long been observed that the size of the nucleus of the cell scales with the cell size, so that this nuclear to cell volume ratio or nuclear to cytoplasmic volume ratio, both are largely maintained. So we will call both things NC ratio for now. Later, I will make a clarification. So here is a picture of different cells with different nucleus to cytoplasm volume ratios. So since we have known that the nucleus scale with the cell as a whole, we are guessing that this NC ratio may be maintained by a homeostasis mechanism, so that if the cell has an aberrant nuclear to cell volume ratio, this ratio should get corrected, like what is shown in this figure. So if you look at the black dashed line, which are regressions of these different paths, the expected NC ratio value should be this 8%. So there is a trend that these different curves are going towards this 8% if the initial conditions are away from that. But on the other hand, if we look at each individual path, there are some fluctuations. So we want to study both this homeostasis mechanism and the fluctuation in this process. Okay, so we started with an osmotic pressure balance model. So here's a diagram from this Devry and Safran's paper. Basically, this is what's happening at the nuclear envelope. So we have these three forces, actually two kinds of forces. So the red arrow and the blue arrow, they represent osmotic pressure. And these green arrows are the tension of this nuclear envelope itself. So these three forces should balance each other, and we have a fixed nuclear size. So, questions?
I didn't understand from the first slide.
Yep.
When you say the volume to nucleus ratio is conserved,
Yep.
it's within the same cell type, or different cell types also have the same ratio?
Yeah, so basically the context should be the same cell type.
Same cell type.
Yes.
When you change conditions, the cell changes size,
Yeah.
but the ratio is maintained.
Yes, that's the idea. So actually, later we will kind of see that for each different cell type, at least according to my model, this ratio should be determined genetically. Of course, for different cell types, we can have different NC ratios. So yeah, good question. Thanks. All right, back to here. This is
that 8%. That was experimental data, right?
Yeah, the 8% in this is from experimental data.
What kind of cells?
Oh, these are yeast cells. Unfortunately I couldn't remember the scientific name, but it's a kind of yeast. Okay, back in here. This osmotic pressure balance model is actually a simple one. So we assume the osmotic pressure is given by the Van Hoff formula, whereas the tension at the nucleus envelope and the cell membrane can be described by this Young-Laplace law. So I need to clarify also here, we are assuming that the shape of these cells are just spherical. And that's why you see this Rn and RCE, which are basically the radius of the nucleus and the cell. So for these two, the first equation describes the force balance across the nuclear envelope. The second one is the balance at the cell membrane. So this Cn is the concentration of solutes in the nucleus, and CCy here and here, they are the concentration in the cytoplasm. The C0 is the concentration outside the cell. So the right-hand side basically describes the osmotic pressure. The right-hand sides are the Young-Laplace law describing the tension of membranes. Questions? Okay, so when these forces are balanced, we achieve a fixed cell volume and nucleus volume. And but if you take a closer look at the solutes, we have, say, different types of molecules. In this picture, we have these big dots and small dots. So those big dots, both red and blue, they represent large molecules like proteins. And whereas these small dots are smaller molecules like ions. So what we assume further is that the small molecules can freely diffuse across the nuclear envelope because the nuclear envelope is actually porous with these pore complexes. So we assume that back in here, the concentration of the small molecules are the same in the nucleus and the cytoplasm, so that their osmotic pressure across the nuclear membrane is the same, so that we only focus on macromolecules. In our case, it's just proteins. So in this first equation, that's still the balance across the nucleus membrane, but there's no small molecules. These are the concentrations of the proteins, Pn, and the proteins in the nucleus, and Pcy, the proteins in the number of proteins in the cytoplasm. For the second equation, which is the balance across the cell membrane, of course, the small molecules do matter. But what we are interested in are these Vn minus Bn and Vc minus Bc, these volumes. So we only look at the first equation. And from the first equation, if we assume then the nuclear envelope is relaxed and the tension is 0, then these concentrations are balanced, which is concentration of proteins in the nucleus equal to concentration of proteins in the cytoplasm. And if you rewrite this balance, we can see that these volume ratios are simply equal to these protein number ratios. We define this as this NCY ratio. So I'll clarify what these Bn and BCY mean. They simply mean the minimum volume, which are the volume of those proteins themselves. So anyway, what this boils down to is that we can study the volume ratio by focusing on the ratio of protein numbers. Questions? Okay.
You may cover that. The volume of protein, I guess, I'm seeing you don't assume that it's negligible compared to the volume.
Yeah, we can assume that, but for now, it's okay to assume they are not negligible. Because either way, here, I see your point. So yeah, I should further assume they are negligible. But I think that the point I'm trying to make is that we can study the volume ratio by studying the protein ratio. Okay, good. Yep, so that's this. Before I move on to the protein ratio, there is this deterministic model we've done before that I need to introduce. So to study this homeostasis, we assumed this exponential growth model of the volume. So the cell volume just grows exponentially. And for the nuclear volume, we just assume the growth rate is this fraction f0 of the growth rate of the whole cell. Then, okay, we can apply the quotient rule to this derivative of the nuclear to cell ratio, which is volume n divided by volume cell. And what we have is the derivative of this nuclear to cell ratio is given by this formula, which means if this NC ratio is greater than F0, then this whole thing is negative and this will decrease. Whereas if this is smaller than F0, then this right-hand side is positive and the nucleus to cell ratio will increase. So this is referred to as a passive feedback mechanism, which should be able to correct the perturbations to this steady state value of F0. Okay. But then again, this is deterministic. There's no fluctuations, so we want to study the model as a stochastic model and look at this from a statistic point of view. So we need a stochastic model for the growth of protein numbers. So this is the model proposed by Lin and Amir in their paper. This looks technical, but the point here is we just assume the transcription rate for a specific gene is proportional to the fraction of RNA polymerases working on this gene, and the translation rate is proportional to the fraction of ribosomes working on this gene. So basically, we have these three processes describing this translation, then transcription, translation, and the mRNA degradation. So if we look at these reaction rates, they are actually time dependent. But what's more problematic is this translation process, where this PR, the number of ribosomes, is time dependent, and also this fraction of mRNAs are also time dependent. So we want to obtain an analytic solution of this model. This is too complicated to do that. So we considered a more simplified gene translation model. So compared to the previous one, this is only the translation process, and we kind of ignored for the moment the transcription. So we have these translation processes where these three kinds of proteins are involved. PR is the ribosomes, PN is the nuclear proteins, PC is the cytoplasmic proteins, and this number KT is the translation rate. And these phi's, they are the effective gene fractions of these three kinds of proteins. So here we are, like the previous model, assuming that the translation rate for each of these proteins is proportional to their gene fractions. Questions? Okay. So this model is much simpler than the previous one. And like I said in the title, these reactions are all first order because these reactants, well, they're only, for each reaction, there's only one molecule. So this is first order. These models are exactly solvable. So then we write down the chemical master equation corresponding to this model. This describes the joint probability distribution of these protein numbers and how this evolves in time. So, okay. Questions? This is the master equation. We want to solve this for this joint distribution. And we need, before we do that, we need an initial condition. So here there's a distinction between the intrinsic and extrinsic noise. So what we're saying here is we want to focus on the intrinsic noise, which is the fluctuation from these reactions. So we set this initial condition to be deterministic so that we can isolate the effect of this intrinsic noise itself from other extrinsic noises. Okay, questions? Yeah.
These models, they diverge in terms of numbers of fractions converge
What do you mean by diverge?
into infinity.
The number of these proteins?
I mean, little p is a fraction, right? So
No, they are numbers, sorry.
So how does it diverge from the Markov equation? Because you have this symmetry, I mean, you have this duplication.
Yeah.
At the rate.
Yeah,
So did you have a condition? I mean, I had the impression when you showed the transitions that it would PR would be.
Yes, that's the case. There's no restriction. This is unlimited growth, and they will actually, if you look at the mean, they will grow exponentially and go to infinity.
Okay.
So, yeah, I know this is not really realistic, but for now we will just look at the simplified model.
And here, the initial condition.
Yeah.
Is there some typo? This should be like PR minus PR prime or something. Is this?
Oh, that's PR comma 0.
Oh, PR comma 0. Okay.
So what these numbers are? Wait a second. What's going on? Oh. Okay, I accidentally hit something. Back. So what are these initial numbers of proteins? We want to study the effect of the system size on the level of fluctuations. So we introduce a system size variable n big N and these initial protein fractions. so that these PR0, PN0, PC0, they are given like so. Basically, they are fixed numbers so that this P at 0 is only a point mass at this PR0, PN0, PC0. All right, so we have the equation and the initial condition. We can try to solve that. And the method is this generating function method. So we look at the generating function of this joint distribution using a formula in Gardner's Handbook of Stochastic Methods. We actually derive a PDE for this generating function from the chemical master equation. And of course, we need the corresponding initial condition. So the point here is this partial differential equation is a first order one. And we can solve this equation for G analytically using the method of characteristics. So yeah, we can write down the characteristics equations, which are a set of bodies. Everything else looks pretty simple except for this equation for SR. But unfortunately, this equation is actually solvable analytically using partial fractions. So anyway, everything now is solvable analytically, and we obtain a closed form expression for the generating function. So yeah, again, this looks pretty technical, but the point here is now that we have this expression, we can calculate this joint distribution using this Cauchy integral formula. Well, in practice, we don't really use this formula numerically. So instead, oh, any questions? Okay. Instead of using this formula, we do a change of variables. We set these as variables to be this e to the 2 pi I theta. And then this Cauchy integral formula just becomes this. What is this expression? This is simply a Fourier transform of this psi function. So this can be efficiently computed using standard FFT software packages. The only problem is if the protein numbers get large, this is pretty memory intensive. So I use a variable size grid based on Chebyshev's inequality to reduce the RAM size usage size. Anyway, what boils down here is we can now calculate the joint distribution. Yep.
So theta is complex here, or did you have modulus of each?
Oh, theta is actually real between 0 and 2.
So how, I mean, so the variable for G were modulus 1?
Yeah, I guess to kind of answer your question, this integral is done well on these unit disks. So yes, this is doable, modulus 1. Anyway, we also tried this spectral Galerkin method, which seeks to represent this joint distribution using this ansatz, and plugging this ansatz into the master equation yields a system of ODEs of these coefficients C, and they can be solved using ODE solvers. So the point of this is, I mentioned that all these methods, the FFT and these generating function, they only work for first order reactions. So in principle, the Galerkin method don't have that restriction. It should work for arbitrary reactions. But unfortunately, this doesn't work very well for our model. So we'll see in this picture. Since the joint distribution is 3D, I couldn't show that in a picture, and I could only show these marginal distributions. So here we can see, well, first of all, these histograms calculated from Gillespie algorithm, and these red curves are the results from the FFT. The blue curves are from the Galerkin method. So we could see that the FFT method gives pretty good marginal distributions compared to the histograms. So the red curve here, this, and also this, they pretty much agree with the histograms. But for these blue curves, well, there's some problem in here, and there's a little bump in here, and there is this kind of ripple thing in here. So that's why I say, unfortunately, this Galerkin method doesn't really work that well. We have numerical artifacts. So I choose to use the results from FFT to calculate our NC ratio, but I will mention that later. Some more observations of these marginal distributions can be made. So the marginal distribution of the ribosomes, which is the first picture here, this is actually a negative binomial distribution. Why? Because the reproduction of ribosomes, this is a pure autocatalytic reaction. The other two reactions doesn't really affect the number of ribosomes. So this result is actually known since the work of Max Delbruck in the 1940s. The other two marginal distributions are not negative binomial, but the production of the other two proteins can be viewed as a Poisson processes driven by this stochastic process of the number of ribosomes. So this is a new viewpoint that we want to make use of later. But for now, these are the observations from these marginal distributions. Okay, still have some time. That's good. So once we have the distributions, okay, we can calculate the NC ratios, but bear with me a little bit. There's still a little more to say about these number of proteins. So we can actually calculate the moment of these variables from that closed form expression of the generating function. This is simply done by symbolic differentiation, so that's pretty convenient. The point here is, yeah, if you look at the mean number of ribosomes, this is indeed an unrestricted exponential growth with this growth rate. So indeed, this will go to infinity if the time goes to infinity. Then the other two also grow exponentially with the same rate after some initial relaxation. So yeah, we have this exponential growth in the mean. And for the variation, for the variance, we look at these coefficients of variation defined to be the ratio between standard deviation and the mean. So all three coefficients of variation are of order big N to the -1/2. This means that if our system size gets really large, these relative fluctuations should become negligible. So, okay, I guess that's all I would like to say about these protein numbers. And we can move on to the calculation of the NC ratio. But here first we have to make this clarification. Like we mentioned before, actually there are two kinds of ratios, nuclear to cytoplasm versus nuclear to cell. They are actually different in this definition of protein ratios. So yeah, note that the ribosomes are actually also in the cytoplasm. So if we calculate the cytoplasmic protein number, that's PC plus PR. And for this nucleus to cell, well, the denominator becomes the total number of proteins. The numerator is still just the number of proteins in the nucleus. So we will focus on this NCE, nucleus to cell, because we did this in the previous work. But as we've seen in the osmotic balance model, it's actually a little bit more convenient to use this nuclear to cytoplasmic. In that case, we could not derive one from each other. We could not derive one from the other, but both can be conveniently calculated if we have the joint distribution. Yeah.
You mentioned in the earlier slide that the protein number keeps increasing.
Yes.
Over time. So what controls that, or is it allowed to go to infinity?
What?
The mean ribosome number that you are showing it is exponentially increasing.
Yeah, it goes to infinity.
So it just keeps increasing, is it?
Yes.
So it's not controlled at all.
Yeah, it's not controlled. Like I said, this is not really physically realistic, but we just want to focus on the simplified model for now.
In the biological case, this would be limited by cell division. So that's
Yeah.
basically what's
I mean,
missing
we
from
also have protein degradation.
Yeah, you could add a protein degradation as well.
But right now it's not.
Yeah, it's not included. And to clarify, I am aware of all these matters. It's just that I want to keep things as simple as possible. Yeah, okay. So we want to actually calculate this ratio. How to do that? Well, we simply view these ratios as functions of these random variables. And once we have the joint distribution, these moments can be straightforwardly calculated. And the first moment is simply the mean. And the second moment, well, we can then calculate the variance from this second moment. So, okay, I need to move on a little bit. So next I'll show some results. These curves are the means calculated from the FFT method distributions and with different initial conditions. So this shows relaxation to this expected value of 0.08. Basically, this picture demonstrates the nucleus to cell ratio homeostasis, but a few observations can be made. So first, of course, the mean curves will eventually approach this expected value if the initial condition deviates from this value. But what's interesting is this this curve in the middle. So it starts exactly from this 0.08, but it doesn't really stay on this as expected. Instead, it goes up a little bit. This is the system size n equal to 100. So this is observed at other system sizes as well. So due to fluctuations, this mean curve may actually overshoot the steady state a little bit. Later in the Taylor expansion approximation, we may kind of see why. Finally, an important point is if we compare these two curves above and these two curves below, we can see that if you look at the rate of change, the further the initial condition deviates from this expected value, the faster this mean curve returns to this expected value. So this agrees with our previous deterministic exponential growth model. So the further it deviates, the faster this returns. This is achieved without any active feedback mechanism. So this is exactly like that previous passive feedback. Questions? Yeah.
I'm not very clear about the 0.08 numberization. So why does it reach that value?
Yeah, this 0.08 is observed in the experiment for this type of cell we study.
Yeah, so what features of the cell have you integrated in the same number as the one for that particular cell type, right? You said it varies from cell to cell, cell type to cell.
Yeah, well, actually, yeah, this number is this 0.08 is this phi n, the gene fraction of the nuclear protein. So yeah, if you change this in the model, these will go to that different values.
No, so was 0.08 an input to the algorithm, or is it like just the output that you're getting out of the model?
It's the input.
Okay.
Yeah. So I hope that clarifies.
And I mean, I think that's basically the only biologically accurate parameter, because the growth rates you're using now are kind of like toy parameters.
Yeah, so
just to illustrate the expected qualitative dynamics, but I think right now the only real sort of input parameter that's based on the biology is this fraction. And it's not really even true that that is a biologically measured parameter, because what's measured is not the gene fraction of the nuclear proteins, but rather just the NC ratio.
yes.
Since we're kind of putting in, we know that if we put in for that parameter 0.08 in this model, that should be
Yeah,
the steady
I was
state.
just wondering how did the algorithm come up with the value corresponding?
No, we're putting that in.
Yeah, that's an
But
input.
then it's interesting that with this, as Xuesong is saying, like when you have the stochastic version of the model,
Okay.
actually it's not quite plateauing at the 0.08, but you get something a little bit different.
Okay, and just one more thing. And is it also known that the protein ratio in the nucleus to the cytoplasm is actually the same? Is it?
From our work, that's not known. We didn't.
So the most of this model is, it's not that we're able to kind of experimentally count numbers of proteins,
Yeah, that's a
numbers of
little bit
soluble
difficult.
molecules, but what the experimentalists are able to do are sort of osmotic perturbations to the cell,
Okay.
and so they can experimentally change the osmotic environment, and what we see is that the cell then swells or shrinks, the nucleus swells and shrinks in
Yeah.
proportion, in a way that suggests that it is indeed osmotic pressures controlling the size.
Yeah.
Yeah, and that's why we have that osmotic model at the beginning.
Okay. Sure.
Yeah, okay, so back to these curves. Previous pictures only look at the mean. Now we can check the variance as well. This picture shows this NC ratio curve with different system sizes and their corresponding standard deviation. So if you look at the mean curve, it's pretty close to each other in this picture. But if you look at the standard deviation, we can see that if we increase the system size, the standard deviation actually decreases. So this is just an example. The problem is due to the memory limitation issue for this current growth rate I choose, which is 0.005. It's only kind of realistic to calculate the distribution up to 2000 seconds using FFT. And then the system size couldn't be too large either. So, but at least we see this trend of the decreasing variation when the system size increases. And we would like to explain why this is happening. And that leads us to the Taylor expansion approximation of this ratio. So we can actually calculate the mean and variance of this ratio using a formal Taylor expansion of this ratio function x1 over x2. So we let this x1 equal to our Pn, x2 equal to this total number of proteins. We can calculate these approximations. But then if we look at these results themselves, this mean, this is expanded to second order, it's actually this ratio of these means plus some other some other terms that actually depend on the variance and the covariance. So here we can kind of see that these fluctuations have some effects in this mean value of this ratio. So for the variance, then it's only feasible to expand it to the first order, and it looks like this. So these are the formulas we use. Like I said, we need to plug in x1 and x2 for these protein numbers to calculate the actual ratio, the mean and variance of the ratio.
So here you're assuming that the variance is small.
Well,
Variance of x2, then assuming variance of x2. Otherwise, this is not.
yeah, actually, I'm not really assuming the variance is small.
But what is the expansion with respect to? What is the?
Oh yeah, the expansion. Yeah, the expansion is actually with respect to the mean value. So yeah, I kind of see your point. This would be not really accurate if the fluctuation or deviation from the mean gets really large. But I just want to use this picture to convince you that this works pretty well in our case. So if we look at these curves, the mean curve, the red one is from FFT, the blue one is from this expansion approximation. They agree with each other pretty well. And if you look at the standard deviation, of course, yeah, the FFT method gives a slightly larger SD, but still, I would say this is a pretty good approximation in our case. So now that we are convinced that this approximation works pretty well, we can actually, now we don't have the restriction of memory usage. So I can explore longer time and larger system sizes. So here I have to show these three pictures because this middle one is exactly the initial condition starting from 0.08. And we can actually see when the system size is small, this overshoot is really kind of prominent. Whereas if the system size increase, this gradually gets flatter and flatter until it's kind of indistinguishable from this flat line.
But still, all of these are less than 1%. Right.
Yeah, I think so. It's not very
The
large.
y-axis is very zoomed in.
Yeah, this is very zoomed in. So this overall, this shouldn't be very large, but just to make a comparison, this smaller system size gives a relatively larger deviation. And this, they are the case where we have initial condition deviates from the 0.08. This should go down, this goes up. But this picture, the point is, again, different system sizes leads to different curves, and they should approach this black dashed line called the ratio of average. So what is that thing? This is exactly what's happening. So I actually have a closed form expression for this Taylor extension approximation, but they are just way too technical. So I won't show them here, but just I want to show that this expansion actually have this form. This first term is this ratio of the means, and the second term, we have a numerator that depends on these parameters and also time. The numerator is simply the system size big N. So for a fixed combination of these parameters, if we increase the system size, this will go to 0, and this whole mean of the ratio should approach this first term, which is ratio of the mean. And this is what I mean in these pictures. When system size increase, these different curves gradually gets closer and closer to this black dashed line, which is that ratio of these means. So it's the same here, here, and also in here, which if we start from 0.08, the ratio of average is a flat line of 0.08. So this is the system size thing. And the ratio of the mean itself, this is a little bit technical, but I need to show that because if you look at this expression, if t goes to infinity, these two grow, and this limit of this whole thing will just be the phi n showing up in here. And this is that NC ratio homeostasis that I'm mentioning before. So questions? Okay, so this is the mean. The variance, well, yeah, I am a little bit over time, but I started later, so I can go a little bit longer. This variance of the NC ratio is this number multiplied by this function, which will go to 0 when system size is large. So the fluctuation of this nucleus to cell ratio should become negligible when the system size gets large. So these are the conclusions that I have. So yeah, we can conclude that using this simplified translation model, we can explain the NC ratio homeostasis via growth. But since we have this result, the fluctuation should become negligible when system size is large. Maybe this model is not really sufficient to explain the fluctuation observed in experiments. Because in natural cells, the number of proteins is really large, like 10 to the 5th, 10 to the 6th, whatever. So according to this formula, we shouldn't really observe any fluctuations in these NC ratio curves, but they are there in reality. So how to explain these fluctuations, that's something about the future work. Maybe we are ignoring something. A more realistic gene expression model with both transcription and translation, it may be useful. But I think what's more important is we only focus on the intrinsic noise from these gene transcription and translation. And there are various sources of extrinsic noise on this NC ratio. For example, when cells divide, these molecules get randomly partitioned. This might contribute to this fluctuation. And the other one, the nuclear transport process, because this model simplifies to, only the protein ratio, we didn't really care about the dynamics. If you look at the transport, maybe there's some noise in there. And finally, this osmotic phenomenon itself leads to some fluctuations that may be interesting to study in the future. So, okay, I guess that concludes my talk. And thank you all for, okay. New questions?
I think we need to go out of the room.
Yeah, so,
Sorry, we do have a class in here.
yeah, sorry about that. I had some.
This is the time block where we only have a